All right, let's jump right in. Today, we've got a real scientific mystery on our hands. It's a genuine viral detective story. We're going on the hunt for a whole family of missing viruses. And forget magnifying glasses. The detectives in this case are using something way more powerful, massive digital databases.
So yeah, that's the big question, right? How do you even begin to do that? It's this wild puzzle that takes us deep into the world of genetics and to some places you would never expect. And the craziest part? It all happens without anyone setting foot in a traditional lab. Come on, let's follow the clues.
Okay, so our story starts with this family of viruses. They're called the Amalgaviridae. And you can think of them like any big family, with lots of different branches. But there's this one branch that was, well, a total mystery to scientists.
Now, this family's got this signature move, a really clever genetic trick they use to build their proteins. We'll get into that a bit later. For now, though, just imagine this huge, sprawling, viral family tree. Most of the branches are packed with relatives, But one of them, it was pretty much empty.
And this is our cold case, the Zybavirus genus. I mean, before this investigation, it was seriously lonely. It had just one single official member, Zygosaccharomyces bolivirus Z. Let's just call it ZBVZ for short. It was this massive question mark for virologists. Like, why is this branch of the family tree so bare? Are there others out there just waiting to be discovered?
So to crack this case, researchers didn't grab a shovel or a test tube. Nope, they turned to this incredibly powerful modern technique. They went hunting inside a computer, digging through absolute mountains of genetic data that's all publicly available.
And this method has a name, transcriptome mining. It's basically digital forensics, but for biology. It's amazing. Scientists can scan for these tiny, unique genetic fingerprints of viruses that could be hiding in plain sight, tucked away in data from thousands of totally unrelated experiments from all over the globe. It's a treasure hunt, but the map is petabytes big.
The way they do it is it's so clever. It's like casting a digital dragnet. First, they take the genetic code of that one known Zybavirus and use it as bait. Then, they sift through millions of hits to find the really promising leads. And for the final step, they take all these little fragments of genetic code they've found and piece them together, kind of like reassembling a shredded document, to build a complete, brand new virus genome. It's incredible.
And boom, this is the moment the case just blows wide open. Because this digital search didn't just find a clue or two, it uncovered a whole lineup of new suspects, potential new members of this ghost family of viruses.
The hunt was a total success. I mean, the team didn't find just one, they managed to piece together 4 completely new Zybavirus-like genomes, basically pulling them out of the digital ether. But the real shocker wasn't that they found them, it was where they found them.
Okay, just look at this list. The variety is just wild. I mean, one was in fermented grains. Okay, that kind of makes sense, right? The original was found in yeast. But then you get to the others. One was linked to a centipede, another to a fruit fly, and a fourth was just hanging out in salt marsh sediment. Nobody saw this coming.
So you're probably thinking, wait, these viruses infect centipedes? Well, the researchers don't think so. The key piece of the puzzle is that Zybaviruses are actually mycoviruses, meaning they infect fungi. So the most likely story here is that the viruses are infecting fungi that just happen to live on or inside these other creatures and environments. The centipede and the fruit fly, they're basically just the getaway cars for the real hosts.
All right, so our detectives have their suspects lined up. But, finding them is just half the job. Now comes the hard part, proving they actually belong to the Zybavirus family. It's time to do some genealogy. It's time to build the family tree. The scientific tool for this is called phylogenetic analysis. What they do is compare the genetic sequences of the new viruses with the ones they already know about. By doing this, they can literally map out their evolutionary relationships and see exactly where everyone fits in. It's basically a DNA test for viruses to prove who's related to who.
And right here, this is where we get the biggest plot twist of the whole story. The analysis comes back, and yep, 3 of the new guys, ZBZ2, CheVZ1, and DisaVZ1, they're confirmed. They're part of the Zybavirus family. But the 4th one, SCMVZ1, it's the odd one out. It doesn't fit at all. Instead, it groups up with a bunch of other unclassified viruses, forming what looks like a totally new branch on the tree.
So just think about that for a second. They went looking for missing members of one family, and in the process, they stumbled upon what could be a brand new genus altogether. They set out to solve one mystery and accidentally uncovered a second, maybe even bigger one. That's incredible science right there.
But wait, there's more. The discoveries didn't stop there, because now, with all this new genetic data in hand, the researchers could finally dig into another puzzle, that clever biological hack I mentioned earlier, the one these viruses use to replicate. And figuring out these kinds of tricks, well, that's how we start to understand how life itself evolves.
So this trick is called programmed ribosomal frame shifting. Here's the deal. The virus's genetic code has this sneaky built-in instruction. It makes the host cell's protein-making machine kind of hiccup and skip ahead one letter while it's reading the code. This tiny little slip lets the virus build a special giant fusion protein. It's like a viral Swiss Army knife. And that protein is absolutely essential for the virus to make more copies of itself. It's literally a typo that's there on purpose.
Now, for a long time, scientists thought they had this code completely figured out. They thought it was always this one specific sequence, UUU, CGN. But when they looked at the new viruses they just found, they saw little differences. For example, that centipede-associated virus, it used UUU CCU instead. Suddenly, the code didn't seem nearly as strict as everyone thought.
So by looking at all the viruses together, the old one and all the new ones, the researchers could see the bigger picture. They figured out a broader, more fundamental code for this hack, UUU_CNN. And what this suggests is pretty cool. It means the original, ancient version of this trick was probably more flexible. And then, over millions of years, different viral families evolved to specialize in their own preferred version. It's like we're getting this little glimpse into the deep, deep history of how viruses evolve.
So, where does this all leave us? What's the bottom line here? We found new viruses, we might have a whole new genus on our hands, and we cracked a fundamental genetic code. Let's just wrap this all up and see what this amazing detective story really means.
So what are the big takeaways? First off, these public databases are an absolute treasure trove for finding new life. Second, the Zybavirus family, it's not a lonely twig on the tree of life, it's a big, diverse branch. Third, this research might have just put a whole new genus of viruses on the map. And finally, figuring out these viral hacks gives us a front-row seat to watch how evolution actually works. And that leaves us with one last kind of mind-blowing thought. Remember, this whole study was a very focused hunt for one specific type of virus, and it found 4 new ones, and maybe even a whole new genus. So it really makes you wonder, how much more of our planet's biology, how many countless new species, families, and incredible evolutionary stories are just sitting right there, hidden in plain sight, in data we already have? This one case might be closed, but you get the feeling the real investigation's just getting started.